Okay, any questions from last week about the syllabus? Now it's been a week. You've had a long weekend to go over stuff, think about the stuff, and kind of get familiarized with some of the topic. I know that the crash course on neuroanatomy happened last week, and some of you attended. If you haven't attended, the recordings are on Canvas. I highly recommend that you should all watch it. It just gives you a nice overview of the material, some of the stuff that probably we covered in the class, some beyond that, but you're not going to be questioned on them. It's just meant to give you a background on the topic and just, just reinforce some of these basic stuff that you may not have been exposed to before or have seen in your undergraduate courses. Okay? Yeah. Is it not? I could actually, yeah, I think the voice is down here and this thing is not helping for some reason. Better now? Yeah? How about now? All right. Yeah, it was the volume was down. So any questions, concerns? No? All right. So we're gonna get started where we left off last time. We were on this slide, and I was talking about how neurons communicate with one another. Prior to that, we talked about the fact that neurons can be either excitatory or inhibitory, and that's property of the neuron itself. So one neuron is excitatory, means that it releases excitatory neurotransmitters. And if it's inhibitory, it means that it releases inhibitory neurotransmitters. And those labels mean something about the way this neuron impacts postsynaptic neuron. So this is a presynaptic cell. It releases different kinds of neurotransmitters, and those neurotransmitters impact the postsynaptic cell. Every neuron, as we were talking about it, has, let's say, some of these pyramidal neurons in the cortex have on the order of, let's say, 500 incoming synapses and 500 outgoing. So it means that they're integrating input from a large number of other neurons and then passing that information to a large number of other neurons. At any given time, the neuron is receiving a lot of input and it's integrating them, and it has a baseline activity. So the way we would visualize this, say, in this case, when you look at this cell, it's getting a lot of inhibitory input, I, and a lot of excitatory input. And when it integrates all of this information, then you would get a firing rate. Each line here shows action potential. So at times, we use this visualization to show the firing rate of a neuron, which means how many times in a second the neurons has an action potential. That's the firing rate. So if the neuron is integrating all sorts of excitatory and inhibitory connection, inhibitory input, its activity will be at what we call a baseline. So it's just baseline activity. The neuron is not dead, so it would have a baseline activity. Different neurons from different parts of the brain have different baseline activity. Some of them could have 1 spike per second, just baseline activity. Some could have more, some could have less, like 1 spike every few seconds. So the neuron always has a baseline activity because it integrates its input from a lot of different places. Now, if the neuron gets a lot more excitatory input than inhibitory input, its firing rate is going to go up. It's just because it's getting a lot of excitatory inputs. This means that postsynaptic cells that are connected to this cell, the ones that are excitatory are now active. So if this is a cell and there are a lot of synapses coming to it, if the excitatory cells here are active, then it's going to lead to a lot of more excitatory input for this cell. If again, excitation gives even more, then you're going to have even higher firing. Now, last year I used the metaphor, and I think it works, which we talked about like gas pedals and brake pedals. Okay, so imagine every neuron being a car with 500 gas pedals and 500 brake pedals. At a certain time point, some of these gas pedals are pressed, some of these brake pedals are pressed, and the neurons has a baseline activity. Now, if you put your, if you press more gas pedals, what's going to happen to the speed of that cell, the speed of that car? Go up. If you even have more gas pedals and press them harder, then it's going to go even higher. So what is the other side of this? Well, what if you actually start pressing the brake pedals more? What's going to happen? It's going to slow down the car. What if you really press all the brake pedals? Well, it's going to almost make the cell silent. So think of every synapse that is coming to a cell as either a gas pedal or a brake pedal. So what are the two? So if a cell is at baseline with that metaphor, what are the two ways that the cell firing rate could go up? Okay, so in our metaphor, what does that mean? It means that we're either pressing the gas pedals or we're picking our foot off of the inhibition or off of the brake pedal. Does that make sense? So there are four conditions here. What are the four conditions? Well, you have more excitation or less excitation. You have more inhibition or less inhibition. So there are four possible ways that the cell firing rate could change. And those four ways, people get confused about less inhibition, right? There is always some inhibitory input coming to a cell, but that inhibitory input itself could get silenced. How would that happen? How would a cell get less inhibition? So first, what does it mean in the neurotransmitter space? When a cell is less inhibited, what does it mean in its neurotransmitter? Yeah. Yes. But what does it mean? We're going to talk about what it means for presynaptic cells, for this cell, right? If a cell is less inhibited, it means what for the postsynaptic cells? Or presynaptic cells, sorry. Yes, but so let's say if I'm saying it's less inhibited, not that more excited, but less inhibited, it means that there was some inhibitory input to this cell. What happens to that cell that was inhibitory? The presynaptic cell that was inhibitory. It's not firing. What does that mean? It means that, okay, so I have a cell here, right? This cell, when I go up here, you're pressing all these brake pedals. It means that there is a presynaptic cell that is releasing inhibitory neurotransmitters. That's the only way it would work, right? So there is a presynaptic cell, it's releasing a lot of GABA. But the reason it's releasing a lot of gamma is that because it's active. This brake is being really pressed. When you're less inhibited, it means that this cell is going quiet and it's not releasing inhibitory neurotransmitters anymore to inhibit this postsynaptic cell. Does that make sense to everyone? Yeah, kind of, right? So there are four, again, people get confused, but think about these four conditions and then it becomes a little clearer. Question?
Yeah, I had a question. Last week you talked about sort of, I don't know if this is the right verbiage, but an action potential failing. What is a case in which, I'm presuming action potentials are a response to a stimulant. What is a case with which you start to have some sort of reaction to stimulant? Don't.
So a lot of time for a cell to have an action potential, it has to get a lot of excitatory input. So let's go maybe to this, right? For a cell to have an action potential, it has to get a lot of excitatory input, very little inhibitory input, for it to eventually reach a threshold that can it explode, right? There's let's say minus 40, minus 45 millivolt from minus 70. It has to go to that level in order to explode. But if it only gets a few excitatory inputs, or the excitatory inputs are not arriving at the same time, then it's not going to reach that threshold.
Right,
So it's going to fail.
Example of something that I see a stimulant, something is stimulating in the environment. Like, not really that.
So let's think these are different scales, right? We're here talking about a single cell. You're talking about a perception, right? So if I'm looking at an object, I'm looking at that pink sticker, right? And then a lot of cells are active. Maybe a lot of cells fail to fire action potential because they were representing a different color or they were representing an adjacent color. But the cells that are really like that pink color would fire, but the cells that are, let's say, signal red color may not fire because that's not really driving them to, you see what I mean? So, but there are a lot of cells. We're talking about a few of them. We're talking about one. How does one behave? But you can imagine there's a collection of cells. Some of them get stimulated, some of them. Okay, so so the basic idea here is that you're getting a lot of input. Every cell gets a lot of inputs. Some of them are excitatory. What does it mean when the cells are excitatory? When the inputs are excited? When I say cell gets a lot of inputs and they're excitatory, what does that mean? Yeah, so so it means that the neurotransmitters that are being released from those cells are excitatory, are exciting the cell. So that's what it means when it receives excitatory input. When I say a cell receives a lot of inhibitory input, it means that a lot of other cells are connected to it where they release inhibitory. But for those connections to actually have an impact, the cell itself has to be either above or below its baseline. It's not that, so when you look at here, so you have the postsynaptic neuron and you have the presynaptic neuron. This synapse could be excitatory or inhibitory, depending on the type of neurotransmitters that are being released. But for this synapse to have any impact on the postsynaptic cell, this cell actually has to fire an action potential or be above its baseline to do something to impact the postsynaptic cell. Does that make sense? So there are multiple things that are involved in it. So there are three factors here. One, what is the activity of the presynaptic cell? Presynaptic cell could be above or below a baseline, its own baseline activity. What is the type of the connection that it has? Is it inhibitory or is it excitatory? What is the strength of this connection? Right? Connection. So two cells could have many synapses to each other. It's not that you only have one synapse. And depending on how many synapses one cell has to another cell, it changes the strength of their impact on one another. So if there are multiple synapses from one cell to another, this postsynaptic cell can really impact this presynaptic cell could really impact the postsynaptic cell. Okay, so there are three factors. It's going to be important because I'm going to go into neural networks and we're going to talk about this a lot more. But the factors are, what is the activity of the presynaptic cell? What is the type of connection they have? And what is the strength of that connection? So based on that, the cell kind of aggregates all these inputs. There, as I said, there are hundreds of gas pedals, hundreds of brake pedals. Depending on the state of those, then you're going to have the postsynaptic firing rate. Okay, let's move on to today's topic. So now that we talked about the basic operations for how cells communicate. Let's think of an abstraction of that and talk about what we call artificial neural networks. These neural networks are everywhere now. You use them. What is a good example of an artificial neural network in your daily lives? ChatGPT, right? ChatGPT is just a big neural network. And there are other neural networks that you interact with every day. Like when you search something on Google, there's a neural network involved in finding the right websites or ranking them. When you use your phones to take pictures and the faces are recognized. And now newer phones have some image manipulation kind of built into them. And those are all neural networks. So it's important for you to understand the basics of them and in what way do they relate to the networks in the brain, because the computation is kind of similar and they're inspired by the networks in the brain. So it's important to see the connection between them. So let's say we have this very simple network. And in this network, you have inputs and outputs. What are the postsynaptic cells here in this network? Out 1 through 4 are 4 postsynaptic neurons. Input 1 through 4 are 4 presynaptic neurons. Why? Because, well, the direction that we're interested in is this, that the input 1 impacts output 1. So input 1 is a presynaptic cell, for example, for output 1. The connections, I'm showing the numbers are showing the connection weights. So if it's positive, it's the excitatory connection. If it's negative, it's an inhibitory connection. So what I said, there are 3 important things. One is the state. One, let's say you have a state of a presynaptic cell. What was that? Okay. Is the state of the presynaptic cell, the connection weights between a presynaptic cell and a postsynaptic cell, and also whether it's excitatory or inhibitory. If I take only those 3 principles, then I can build a neural network where it takes a pattern of input and then it projects them into outputs. Now you could imagine something like the ChatGPT is a much more, a massive neural network. This is just a toy example that we're going to work through together to understand these principles. Okay, these are at the bottom, these are the state of this network. You can imagine these are the states of photoreceptors in the eye. These could be pixels of an image. But for simplicity, we're showing them positively if they're above the baseline activity and negatively when they're below the baseline activity. So it means that this cell is firing above its baseline activity. This cell is firing below its baseline activity. Okay, so let's work this through. I want to look at, for example, output 1. Okay, so the output 1 from input 1, the connection weights is 0. So it means that it's not doing anything. So it has no impact. That connection doesn't exist. Input 2 is also as a connection 0. So that doesn't also exist. Let's go to input 3. Input 3 is connected to output 1 with a connection weights of 0.15. First of all, is this excitatory or is this inhibitory? Excitatory. So it means that if input 3 was really active, it would increase the firing rate of output 1. Well, what does this negative 1 mean? It's not firing. So it's below its baseline. So what is the net effect on output 1? The net effect is that the response of the output 1 goes below this baseline. In our metaphor, so if this is a excitatory connection, let's say it's a gas pedal. But when this is negative, it means what? For the gas pedal, what does it mean? What? You're picking your foot off the gas. That's what that means. So imagine, because I said every cell has a baseline activity, it means that every pedal is almost a little pressed at any time point, at the baseline. Now it's just a matter of picking your foot off of it or pressing it harder. Make sense? So let's look at input 4 to output 1. Is this a brake or is this a gas? It's a brake. 1 means what? You're pressing the brake. So what is the net effect on output 1? It's inhibitory. So with this type of input, which is the state of these neurons and these sorts of connection, you would get this pattern for output 1. It means that collectively this input and these connections are inhibiting output 1. Does that make sense? Let's look at a positive example. Let's look at output 4. Output 4 gets input from input 1, and the input is excitatory. And input 1 is firing above baseline. So what is the net effect on output 4? It's positive, right? Because what does it mean? It means that input 1 is a gas, basically, for output 4. And it's positive, so it's above a baseline. So it means that it's releasing a lot of excitatory neurotransmitters, and the net effect on output 4 is positive. Yeah.
Here with the diagram, I see a 0.5 and a negative 0.5. I'm assuming the negative 0.5 is inhibitory. So you've got 0.5, that cancels out, and then there are 2 zeros. How do we get 1?
Well, for output 4?
Well, oh, I'm sorry. I'm looking at the, I think about input 1.
Well, input 1 is feeding to output 4.
Oh, okay, I'm looking in the wrong direction.
Yes, so these are presynaptic neuron activation at the bottom. Okay. Yeah, so these are presynaptic activation, whether they're either above a baseline or below a baseline. And we're looking at their effect on the output. 0 in this condition, here it means that there's no connection. But because I drew all the connections, I just wanted to say the 0 means there's no connection. Just ignore this connection.
I have a question.
Sure. These are different outputs, right? And each output has its own connections. Each cell has its own connection, right? So let's work it for output 4. Output 4 gets input 1, which is the state 1 times the weight. Okay? All right. Let's look here. So let's work this for output 1. Output 1 gets 4 inputs. Okay? This is 0, so no impact. 0, no impact. Input 3 is 0.5. It's the connection weight times the state. So it becomes negative 0.5.
Does negative 1 there not mean that the action potential failed?
Negative 1 here is a statement about the firing rate. It's not a statement about a single action potential. It means that this cell normally, let's say, fires 5 spikes a second. But right now, it's firing 1 spike a second, for example. So the firing rate is down, not necessarily 1 action potential.
Okay.
Does that make sense?
Yeah.
So the numbers are really it's a statement about whether this cell is firing above its baseline or below its baseline. Is there a question? Yeah. It means that it's below a baseline. So for example, you have a friend who posts 5 images to Instagram every day. And then let's say for a week, they only post 1 image to Instagram every day. So their firing rate is down. Does that make sense? What's that? Which connection are we talking about? So let's go with our metaphor of gas and brake pedals. If it's 0.5, it means that it's an excitatory. It's a gas pedal. But what happens if you pick your foot off of the gas pedal? What happens to the car? It slows down. So that's the idea. That this cell has an excitatory impact, but if you're not pressing on the cell anymore, the downstream effect is negative, right? Does that make sense?
Sorry, how do you calculate that? My brain might.
You just multiply the state of this unit by its connection weight. So the input 3 has a state -1 times 0.5, and it becomes -0.5. Just a simple multiplication of a -1 by 0.5. Here, you're multiplying 1 by -0.5 and then adding all that up. So technically, what we did, again, in our metaphor, is that we picked our foot off of a gas pedal here, and then we pressed on the brake pedal. Does that make sense? Why that makes sense to everyone? Because input 4 to output 1 is a brake. And then you go above a baseline. It means that you're braking, you're pressing a brake here, and then you're picking your foot off of a gas. Together, they really slow down output 1. So it goes below its baseline. Yeah?
Just to clarify, what is the next one at the top? Like, they're all added together.
It means that output 1, the cell is firing way below its baseline. That's what it means. It means that, as I said, let's say you have a car, you have multiple gas and brake pedals. The car is going 20 miles an hour. If you pick your foot off of a gas pedal, then the car slows down. That's a negative effect, even though it's a gas pedal. Even though it's a gas pedal, whatever you do to it, it could actually slow down the car, right? It's not confusing. A brake pedal, if you pick your foot off of a brake pedal, it could speed up the car. So it doesn't matter if it's excitatory or inhibitory. It really matters whether the activity of the cell before that, is it an excitatory, is it above a baseline or below a baseline? Right? So in a network, there are 3 things that I said are important. The activity of the presynaptic cells, the connection, whether it's excitatory or inhibitory, and how strong it is. So those things impact how a pattern gets transformed to another pattern in a neural network. You could imagine a version of this happening in your brain, where let's say you have photoreceptors, and they have a pattern of light on them. This pattern of light gets transformed into another pattern of neural activity that becomes transformed into another pattern of neural activity, and after multiple layers, it becomes a pattern of activity that is like a face, that you perceive as a face. This is the basics of neural computation. We simplified it here to very simple connections and stuff like that. But imagine a more complex version of this happening in your brain. So in the brain, there are only connections and activations. There's nothing else, right? So where are all our memories and those sorts of things stored in the brain? Where are they? What's that? Not necessarily, like that's part of the system, sure. Sure, but in terms of a network, where are they stored? If I said this is all there is in your brain,
It's an output.
the output is an activation state. How many other things are left in this network that you could guess as the place that all your memories would be? What's that? Yeah. So it's in the connections. So there's no other place left, right? It's a network. There are connections and there are activations. And I'm showing both of them here. I'm showing what the weights of all these connections are, and I'm showing what the activations of all the units are. What are the activations? What do you think activations are in your brain? These are different states of the network. These are different states of the brain activity. Any emotional state, any perception has to be a complex pattern of activation of these neurons, right? It can be there's nothing else. There is a pattern of activation. It gets transformed to another pattern of activation. It gets transformed to another pattern of activation. So you have these patterns of activation in different brain areas, and they're connected through these weights. So the memories, everything that you've learned, all the faces that you remember, all the trauma that you've accumulated, these are all in these connections. They're all in the connections between neurons. Where, for example, we talked about ChatGPT or we talked about other face detection systems. When they learn something, when they're trained, what changes in this system? So when you train a network to do something, like ChatGPT, you're training it to predict the next word. That's basically what is trained word, and then that's how it generates so-called auto-regressively a sentence or a bit. So where are those information stored? The same answer. Where is it stored? In the weights. So when you train a network over time, you're actually just tweaking these weights with some smart algorithm. Just tweak these weights. So when you give it, let's say, set a sentence and saying, can you please complete this? And then it predicts sentence. You just feed in the sentence. It codes them as pattern of activation, and then it transfers them to new pattern of activation, new pattern of activation, and then eventually the output gets transformed into another word. So the essence of it is basically this. Does that make sense? What kind of computation we have here? So what is so different about this, let's say, from a laptop computer? Like we use the computer metaphor a lot, but there's something so different about this than a laptop computer. Yeah. The connections are not fixed. Yeah, that's a good point. But well, let's say the nature of this memory. Yeah. Yeah. Okay, maybe at some point we'll have, you know, humans in unorganic matter. But what is it about the way that this computer works that is so different than a laptop, for example? Yeah.
About like the way to process information, they're like predetermined, therefore default processor, and these neural networks you can play with different connections. You know, laptop processor, you cannot do that. It's for default, and that's it.
Sure. Yeah.
Like there's more room for error.
Room for error. Sure. Yeah, they're more stochastic. That's a good point. Yeah. Maybe somebody could argue that spiking is a form of binary computation, right? You could say in this system, obviously, we don't have a spiking. We're kind of summarizing just above a baseline or below a baseline. But that's a good point, right? It could be that some people would argue that these computers are working with these rate coded. That's what we call them, rate coded, because we're just saying above a baseline or below a baseline. But in a computer, you have transistors that have states of zeros and one. Yeah. Okay. Yeah. Yeah, that's a good point.
There is.
What's that?
Some people argue there is no difference.
Sure, there are some differences though. Yeah. Yes, maybe. I don't know. But about the computation I'm talking about the way the computation and learning happens. Way slower, yeah. Way slower, but far more parallel in a lot of it, right? In a computer, a lot of time you can only do computation one at a time. But in the brain, you could do it in parallel. Yeah. What would be a connection in a laptop, for example? Not necessarily. I don't, yeah. I see where you're going, but not necessarily. I'm gonna take a guess? Yeah. You could have it happens in the brain too, right? You get some inputs and you produce an output. I don't see how that's different from this system. Well, the thing is with these, yeah, so a neural network is equally, like these big neural networks are equally vague in their processing, meaning that you know what's happening, you know all the weights, but it's hard to explain how they arrive at the conclusion because it's, some people call them a black box. They're just too big for us to appreciate and comprehend. And that's just the future in some ways, that we're able to create things that we won't necessarily understand fully. And sometimes you just got to give up on understanding fully something that is too complex, you know, like you just got to be happy with some degree of being able to predict the behavior of a system. Did someone want to take a, yeah, last crack at? Yeah, that's a big difference. That's the biggest difference here. In the brain and in a network, all the information that we know that is stored are in these connections. There is no difference between computation and connection, meaning that you have connections that do compute things based on what you've learned before. So they're all together in a network. There is no separate memory. A hard drive that you have on your laptop or any so-called von Neumann computers is that you have a hard drive and you have memory, you have like RAM, which is faster memory, and then you do computation on it, and then you could write it to that memory again. Then you do some more computation, then you could write it to that memory. There's a separation of memory and computation. So you have a CPU that he has a very fast memory on it. Then you have the slower memory, which is your hard drive. But there's a massive there's a separation between them. In a system like the brain, all the connections are carrying some memory, right? So if I'm looking at the face and then I get a perception of the face, or I get a perception of I've seen this face before, it all happens in these connections, right? They're just transformed through these connections to a pattern that to me means something. To you, it means something else. But to me, it means that, okay, I've seen this face before. But it's all that happens in the brain is pattern of activation getting transformed through these weights to other patterns of activation, and then they get transformed again. And again, these patterns mean something. If these patterns of activities are in a specific brain area, to you, it means the word duck, for example. To someone else, means something else, because brains are specific in that sense with your experience. We cannot decode the same thing from two brains. Um, so, so this pattern of activation could mean something. Sure, yeah, we do extend our memory with these tools, right? A lot of time with the notes and the stuff like that, we do extend our memory. But the computation and memory are not separate from each other. That's what I was saying. That any computation that happens in the brain is driven by your experience and your memory that you've learned. Like these connections carry that information. And as I said, for example, something like ChatGPT, you would think it knows a lot about the world, right? When you talk to it, but it's all stored in the connections. It's all stored in the connections. When you give it a pattern of input, it transforms it to something else, and then eventually it produces the output. But what takes the input and produces the outputs are all these connections across layers. So it doesn't have an external memory that it would consult, or a lot of them are not connected to the internet. So they're kind of memorized the internet in their connection weights. The connections are usually in those models are called parameters. And here we have 16 parameters, and some of them are 0. How many parameters a large language model has? Someone wants to take a guess? Billions, right? There are some of the bigger ones go even close to trillion parameters, right? But let's say GPT-3, I think, had maybe around a couple of 100 billion parameters. So there's a lot of parameters for you to store a lot of information in. In the brain, we talked about people estimate that there are like tens of or 100 billion units, and then there are 100, maybe trillion parameters. So these models are still, even the biggest ones are like 1,000 times smaller than a brain and than a human brain. Does this make sense to everyone? This is going to be on the exam. You're going to get a network, you're going to get a pattern of input, and then you could calculate the pattern of output. It's very easy. Yeah. Oh, I said parameters. In a connection weights are like parameters in a network. They call them. Yeah, if it's a neural network, people talk about them, they call them parameters that you learn. No, not these are activations, right? I'm talking about these connection weights. These are activations. These are activations or a state of these 4 neurons. Yeah.
Could you just reiterate the hard drive part? Just kind of.
Sure. So I was saying in a computer, right? If you do a calculation or if you ask something from it, the computation happens. You move some memory into your CPU, and then you do computation, and then you write it back.
It like
yeah.
The hard drive gives you the output at level brain to know their memory.
Yeah, something like that. Now, you could argue that maybe brain has some functional specializations of hippocampus being some sort of memory, and then the rest of the brain kind of looking up there and stuff like, or replaying some of that. There are ideas around it, but there is still not separate the way it would be in a computer, right? There's still.
So like ChatGPT has parameters, but then it also has like a hard drive.
It doesn't, no. It does not. It only has parameters.
Like my last question, but what is like an example of something that has a hard drive machine?
You could, if you want, you could connect ChatGPT to the internet. And it could go and it retrieve Wikipedia pages and summarize those, and or if it's not on, if it's unsure about something, you could connect it to the internet. So you could potentially build systems where not only they have these connection weights that they've learned certain operations, in those connection weights, you could also learn a way of searching an external memory or a resource. So some of these models are connected to the internet. Like if you've ever talked to the Bing chatbot, I think Bing is connected to the internet in a sense that you ask a question, it has a lot of connection weights, but also somewhere in those connection weights, it could decide to search this on the internet and then summarize those inputs. But it's all just connection weights again. Like in a human, if you ask him something and then they decide that, okay, I'm uncertain about this, let me look up the internet, something like that. No, it doesn't happen in that way of I'm anthropomorphizing, but it's not like that. It's like more of a fixed process where you create a pattern of activity, you turn that into a text, you put that on the internet, you get the output. It's like a much more deterministic process rather than them deciding, oh, I need information. Yeah.
Question. So the inputs are the presynaptic cells, the outputs are the postsynaptic, the lines are the connections, synapses, and then what's the 0 and 0.5?
0, I was saying 0 is like, imagine this connection doesn't exist, like it has no impact. I could remove them. Is that the zero you were asking
No,
about?
no, just like, in terms of a cell, what is zero, negative, of the whole, all the black and yellow circles? How would I label that?
You wouldn't. I would let you know what they are. So the zero, so let's say I give you this input pattern.
What does the number represent? It's not like,
What does the number represent other than its value? Yeah, connection weights. Is that what you're asking? Or
I guess I don't understand what it has to do with like the previous slides and the action potentials.
okay, so we're going a step beyond that, right? Meaning that action potential is I was saying it's in a much more millisecond level. You could see that the cell is firing. But now we're talking about firing rates. So again, the brain has many time scales. If you look at it on a millisecond time scale of the behavior of a cell, you could say, well, it has an action potential there, it has an action potential there. But we could go a higher level of a scale and talk about, okay, what is the firing rate of this cell across 1 second? How many action potential did it have in 1 second? Does that make sense? And then now we're looking at that level. We're saying, okay, what is the firing rate of this cell? What is the firing rate of that cell? Action potential is on a more finer time scale. So we're looking at a different time scale of a normal behavior, of a network behavior. Does that make sense to everyone? Around the base, what does it mean? If it's zero, if I say it's zero, if I say the state of this input is zero, it means that it's firing at its baseline, which could be, let's say, 5 spike a second. I don't know, different cells have different baselines. But in this case, because we're simplifying, we're trying to build this into a machine. So we simplify some of those biological details. And we just simplified it to this level. And we're saying, well, at this simplified level, let's have 3 values, 3 states for a cell. And then it would be 1, 0, -1. It could be either something like that, discrete, or it could be a continuous value. But we're simplifying it. And at this simplified stage, you could talk about firing rate of these cells, and then how they're connected, and then the outputs. Yeah.
So this model is basically a binary model.
Mhm.
So well, like there's another model like a quantum model, for example, where
I don't know about quantum models, but let's just stay with this for now. This is a simplified network where what we're saying is that there are networks in the brain, and they take a pattern of activation through the weights, they become a new pattern of activation. A pattern of activation means what? Means activation across many cells. So you could say, well, in this brain area, for example, a brain area for face processing, the cells are responding this way. And then that could change through weights and become a different pattern of activation in another brain area because they're connected through these weights. I just want you to have a mental model of what it means when we talk about pattern of activation in the brain changing across layers. Yeah.
And it's the wrong way, but it does seem like all the numbers cancel each other out. So where are you getting the 1 and -1?
So, which, for example, output 1,
0.5 and -0.5, so just cancel out.
but the state of the cell matters,
Okay.
right? If it's a gas pedal, or these are connection weights. Connection weights by themselves don't tell you what is the impact of this cell on another cell. You have to have the connection weights, but you have to know what the presynaptic cell is doing.
Now I understand. Okay, thank you.
This is just the connection. On the line, I'm showing the connection weights, and whether it's positive or negative, it means that whether it's excitatory or inhibitory. But then the current state of this cell matters. You can't just, you have a pattern of activation, which means that the current state of the cells, and then they become a new pattern of activation through the connection weights. Work this through, you'll figure it out. All right. So
Actually,
you,
I had a question.
yeah.
So you were saying we have to count on the exam.
Yep.
To be able to get the negative one and agree like that.
Yeah.
Yeah, okay. What is that telling you?
About
Anything. The brain, right? Like I can do the math. Doesn't mean I understand what the math is telling me.
yeah. So what it's telling you is understanding the computation, just getting a mental model of what computation could happen in the brain, right? That when you have a pattern of activation and you have weights, what does it mean? What kind of computation those patterns of activation can go through those weights and then become a new pattern of activation. Just giving you a mental model. We're going to talk a lot about computation and brain areas, basically detecting different things in the image or detecting different things in sensory input. And that really means that different units in the brain are doing certain kind of computation.
So it's not like whether or not you hear the word duck.
It could mean that, right? So we build models to better understand something. Sometimes we build models to build those things in a machine, right? So models can have multiple purposes. It could be, so if it's in a real brain, right, and you have photoreceptors in the eye, certain pattern of activation would happen, for example, if you have a letter A. If I'm looking at letter A, I would have a certain pattern of activation in my photoreceptors. And that pattern of activation becomes another pattern of activation in another brain area in the visual cortex, and then it goes, and we'll talk about all of this. But that pattern of activation got transformed. In this case, this is obviously a toy example. This is not, this is just showing you the process. Then you'll imagine what this process means when we actually talk about different brain areas and visual perception. But at this point, it's about just getting a mental model of what does it mean, what computation happens in the brain, and how do activations get transformed from one pattern to another. It's about understanding computational basics. All right. So when we have different input vectors, we could have different output vectors in a network, right? So it's the same network. As I was mentioning it, for example, in ChatGPT or other networks that you interact with, it's the same network. It has the same weights after it's trained, after you tweaked all the weights in order to do what you want it to do. You could give it different types of inputs and then get different types of outputs in the same network. That means that if I'm looking at a face, I would get a different pattern of activation in my infratemporal cortex, which we'll talk about, than when I'm looking at a picture of a house. So activations in the same network, different inputs leads to different patterns of outputs, and those patterns of outputs are different perceptions. And the reason that happens is that because of these connection weights. And as I said, I'm showing a toy example here with only one layer. In the brain, there are multiple layers of this happening. When you go from photoreceptors or from some input to your auditory cortex to the actual perception, it takes multiple stages of processing in order to transform that pattern of activation to a pattern of activation that means something to you. Okay. So 6:50. Let's take a 7-minute break. Back here at like 7:05. All right, so this is a struggle every year. Like when we go through these topics, I feel like there's a lot of confusion in the class, and we do it every year. So the point of it is that a lot of you may be uncomfortable with these topics, like the way I'm describing it. Um, the way I'm describing it, but that's a good thing. It's a good thing to be uncomfortable with some of these topics because a lot of you, I feel like, don't get exposed to thinking about systems that way, right? The systems as computing something. Um, and and a lot of you, even if you have a neuroscience background or you go through this program of clinical neuro or psychology and everything, and you don't never get to really think about the brain as a system that computes something, that it gets a pattern of input, how is it connected to other systems, or what's a model in a sense that you want to understand a more complex system with a model. So it's good that you think that we have this lecture, and it's good that you think about it in this way. I think it helps you in terms of thinking about other systems that are out there in the world. Um, so, um, but we have, I think we have some stuff that I think last year, I think the TAs put together for getting you kind of more comfortable with computing these things and stuff like that. So I think they can share it with you before the exam again. But there is another question about how are these things related to each other? Well, brain, part of the complexity of the brain is that there are different scales that you could look at, right? Like physics, you could look at quantum dynamics, right? You could look at Newtonian dynamics, you could look at much bigger. So there are a lot of scales that you could look at a system. And you don't want to describe a system at a level that is not useful at that moment, right? If you're interested in how, a baseball player catches a ball, you don't care about the quantum dynamics of that ball, right? You care about how in the physical world is moving. So the right level of analysis is really important to understand the system. Brain has many scales. You could look at the behavior of ion channels. You could look at action potentials. You could look at a higher level, maybe the firing rate of the cell. You could look at the entire brain area and how it behaves across seconds. You could go all the way to looking at development of an individual across years, or you could look at evolutionary time scale, right? So a system has many, many different times. It changes, its behavior changes at many different times scale. And I think part of the confusion is that I've switched from a time scale that I was talking about in the previous lecture to a different time scale. And I think some of you are confused by that. So when I was talking about this behavior, right, I was talking about millisecond time behavior, where an action potential is fired and then it releases neurotransmitters. And a cell, whether it's excitatory, whether it releases, GABA, like inhibitory neurotransmitters, or other excitatory neurotransmitters, it has the same behavior. It fires an action potential. It's just the impact it has on the postsynaptic cells that is different. This scale is millisecond scale. But then now we moved on talking about a slightly coarser time scale. Now we're talking about firing rate. We're talking about across the second, what is the cell doing? How many times is it firing an action potential? Does that make sense now? Like, I feel the scale is different now. We switched. And as the course goes on, we're going to go higher and higher in the scale. We're going to talk about bigger scales as we go on. But we kind of, after the first week, we kind of stopped talking about millisecond level scale. Millisecond level it's important to understand the behavior of a single unit, at the ion channels, the firing of an action potential. It's important on that level. But now we moved on to thinking about, okay, what happens across a second? How many times is the cell fire? And when we build these artificial neural networks, the reason it's important that we talk about this is, one, there are a lot of networks in your world, and it's important that you understand the basics of, you're going to be educated individuals. It's important that you understand the tools that you use. But also it's important to understand the system. Again, I'm going to repeat that word, the computations that happens in a system and how things get transformed from one types of activation to another. I'm going to talk about this one last time. So hopefully there's no confusion after that. If I have a bunch of cells, they have a pattern of activation. Some of them are firing really hard, some of them are not. And that's a pattern of activation, we call it. That pattern of activation could mean something, right? If you're looking at a face, you have a pattern of activation in your fusiform face area. It's a brain area, it's down here, and it has a certain pattern of activation. If you've seen this face before, then that pattern of activation means something else, then you've never seen this face before. But it means something. It could mean a new face, it could mean a familiar face, it could mean your grandmother's face. But eventually, it's a pattern of activation. How did that pattern of activation come about? Well, it was initially just some pattern of activation on your photoreceptors. That pattern of activation on your photoreceptors became a pattern of activation on your LGN. It became a pattern of activation in V1. It became a pattern of activation in V2, then V4, then posterior IT, then anterior IT, then you got to fusiform face area. And we're going to talk about all those brain areas later. But what I'm saying is that this is how a pattern of activation in a certain brain area comes about, through a cascade of processing. Now, this is just to give you a mental model of what happens in a smaller scale if you only had 2 layers and we were looking at the transformation of activation from one layer to the next. You can imagine this pattern of activation could mean something, and I said the word dot, for example, but it's a model, it's a toy example. This is a pattern of activation for these 4 units. Each of them is a cell. They're either above a baseline or below a baseline. The connection weights by themselves can't tell you what the impact is. You have to know what the cell itself doing. If it's not firing, or if it's below, if it's on a baseline, it's not going to impact the postsynaptic cell. If it's above a baseline and it has a positive weight, it means that it's excitatory and it's firing, then it should have a positive impact on the output. If it's negative, means that it's below a baseline, but it was excitatory, it has a negative impact on the output. Because, well, it's excitatory, but it's not firing, and it's below its baseline. So the net effect is negative. So, as I said, there are 4 possibilities. Either the cell is above its baseline or below its baseline. Either the connection is positive or negative, excitatory or inhibitory. So those 4 possibilities, think about that, it becomes clear. Okay, something about the brain is that all the states that mean something to you are distributed. It's not, you're not perceiving activity of one cell in the brain as your grandmother's face, right? There's a theory about grandmother neurons and stuff like that, but it's a collection of cells across a hierarchy in order to give you that perception. Every perception that you have is the result of an activation of a host of neurons, of a lot of neurons active together in a coordinated pattern for you to perceive something. And that's what we call a distributed representation, right? As a result, representations are distributed over all the nodes, and the network can hold many associations. It means that you could have many different inputs and then get different outputs. If you hear a word and you hear another word, they're all going through your brain. They're all going through the same network, but they lead to different perceptions because of these weights, right? There's an example here. If you get this input pattern, you get that output pattern. If you get a different input pattern, you get a different output pattern. So a network can transform this different inputs to different outputs. And that's what a memory is. The memory is this in connection weights. And the reason you hear one word and you associate it with a different image, and you hear another word you associate it with a different image, is that the network is processing them same weights, but those weights take those inputs and transform them to different activations. Okay, so that kind of makes sense. It's not showing well, but the idea is that the cascade of activation in a neural network across layers, you could think of them similar to the cascade of what happens in the brain. And this is something I showed a perceptual cycle before. Imagine you open up that cycle in a way. You have sensory processing, You have attention, so you attend to something, you recognize it, you could put it in your short-term memory, you could put it in your long-term memory, and you could imagine somewhat this sort of cascade, not exactly, but this cascade kind of could map onto a neural network, where initial units, we'll talk about this a little bit more, where initial layers, you're processing certain inputs, then you're processing them differently, and then processing them differently. This is a feed-forward kind of theory of brain function, and it's very influential in a way that, well, you have an input, and then it goes through a cascade of processing in order for you to have a perception. And I'm kind of talking, this course kind of focuses on that perspective. Okay, any questions? Yeah. Information processing. What title? Oh, feed forward. We'll talk about feed forward network later. But basically feed forward means that inputs is being fed and then that forward looking, like just basically feeding forward rather than top down or recurrent stuff that we're not going to cover in this class. We're just going to talk about this perspective of feed forward. Yeah.
Middle layer necessarily hidden, or is it just implying that it's so complex?
Technically, nothing is hidden. So in a network, it's all visible, but it's just terminology that people use. They're saying, oh, there's an input, there's an output, and you have multiple hidden representation. It's not really hidden. It's just a terminology term. But in a network, it's all visible. Like if you build a network that does a task, then it's all visible. You have access to all the activation in the middle too. In the brain, to some extent too, you could record from all brain areas. So you kind of have access to activation in middle areas too. It's just that it's much harder, and we're going to talk about some of the brain recording and stuff now. But it's hard to interpret them sometimes, as you'll see. Like when you build a system to mimic the behavior of a much complex system, it also itself becomes complex. So that's part of the problem. Questions? All right. So we're going to talk about some different techniques for neural recording and for experimental design in neuroscience, because we're gonna see them as you read papers and as we go through this course. So it's good that you're familiar with it. So single cell neurophysiology, it's really about recording activation from single cells. Brain is complex with, as I said, 100 billion neurons, let's say. But you could actually record from a single cell and see what it does. I showed a video last time where we were actually listening to a single cell and what it was doing. We'll see a little bit of that. But this was, a lot of this research was done, I mean, neurophysiology is still very popular, obviously, but when it started and people thought, oh, this method actually makes sense, was maybe in the 50s. And what they did is that they took an animal, and a lot of cases, the animal was anesthetized, so the animal was not awake. And then you create a chamber, you lower an electrode, and then the electrode sits right next to the membrane of the cell. So when the potential goes up in an action potential and then it comes down, you could record that. You could record the activation. And then you could feed that activation to an amplifier. You could either visualize it or you could listen to it. You could connect it to a speaker and then listen to it. And then you could just play around with it. You could play around what you show, where in the screen you're showing it, but the monkey is sitting or the cat is sitting, and then the screen is in front of them, and then the patterns of lights are changing on the screen. So this is the screen that the animal is looking at, and they're recording from a single electrode. And they're going to see how this electrode, they're recording using a single electrode from a single cell. And the dots that you're hearing, those are the action potential. So the frequency of that dot is the firing rate. You could see when the light comes on, this cell fires a lot more, right? So this cell, one cell in the brain, like before this type of research, people wouldn't believe that you could find cells that had interpretable functions like that, right? It's like there are 100 billion neurons and you're recording from a cell and you get such a nice behavior. That was surprising. So what they're doing, and you could see that the cell only responds in a certain area. When the light moves away from that area, the cell doesn't respond anymore. So this process is, but what they're really looking for is finding the so-called receptive field of the cell. So the area of the visual world that the unit responses could be impacted by the light. So this is the receptive field of this specific cell. But it was interesting, like a lot of this early work in the 50s, I'm gonna go here. This was done by Hubel and Wiesel, and they actually won a Nobel Prize for it in 1981 for being able to kind of trying to understand what the visual system does using these experiments, like recording from single cell and seeing, oh, there is some discernible patterns of activation that you could actually interpret that, oh, when I press, you show this light in this part of the image, the cell responds. And we're going to talk a lot more about the reason for that and how the anatomy of it all makes sense. Wiesel is still alive, and he's actually in New York at Rockefeller University, and they're doing a celebration of him. I think in December, he's turning 100 years old. So what you could do is that you could show different stimulus, different stimuli, and look at the firing rate of the cell. Each line here is a firing of the cell. And then you could just turn this light around, right? They could play around a lot with this light. You could make it bigger, you could make it smaller or fatter or oriented in different directions. And then see what the firing rate of the cell would, and then you could find the stimulus, the visual input that really activates this cell, that the cell has a really high firing rate in response to that visual input. If you find that visual input, then you could say, well, this cell actually codes for that, or this cell detects that. You know, people use very different terminology to describe this, but they all mean the same thing. It means that this cell, when it has this specific visual input in its receptive field, it fires a lot. You could say, well, maybe it's signaling to the rest of the brain that this thing exists, right? Every cell could be like a detector in that sense. Yeah, so as I said, this was interesting to see where you could look at single cells and kind of interpret their behaviors. Well, there could be something in this receptive field that is inhibiting it, for example. So we'll look at those later, where different pattern of light could inhibit a cell and it would go quiet. If it's at its baseline, it means that it's not impacting this. It's always important to know that there is a baseline activity. Yeah. What are the what? What is the receptive field? What are the factors? So what makes a cell respond in a certain way? What makes a cell respond in a certain way? Sure. Yeah.
Like what the stimulus does to the survival of the animal.
Yeah, on a bigger time scale, yes. So survival drives a lot of things in our behavior, right? The fact that you detect a certain things, it depends on whether it's like we're all very good at detecting the snakes, maybe, and that's why because we, or historically or evolutionarily, they were our, predators. So it's important for us to be able to detect them. But I'm talking about a single cell. What makes a single cell? So I was saying let's say the visual cortex is in the back of the brain. What makes that, but the back of the brain is so far from the eye. What makes that a visual cortex? They're recording from the back of the brain. Yeah. Yeah, exactly. So any function in the brain is a result of connectivity. If you're functioning in a certain way, it means that you must have been connected in a certain way. So back of the brain is the visual cortex because the input from the eye goes to LGN and then that goes directly to back of the brain. So back of the brain, because it gets visual input, because it's connected to the eye, it becomes the visual cortex. It processes visual information. Why a receptive field is a certain way? Well, let's say the receptive field of, let's say I have a cell, the receptive field is here. Well, it means that the photoreceptors that get input from this part of the visual world must be connected to this cell somehow across multiple layers. So it means it's every function in the brain is a result of connectivity. And it's an important point to internalize that you have a function because you're connected in a certain way. We'll talk about that in a lot more details. That, like, we'll talk about this, the response of different type of cell and how that response comes about. What are the detailed connectivity that gives you that specific response? So that's a great question. I guess forward-looking question. But the fact that these interpretable responses exist themselves, that's an interesting finding, right? That the fact that you could actually play around with some patterns of light and then find very specific area of the visual space with a specific orientation of light that gets you the response from this cell. I think that's interesting, right? Now you could actually move that electrode, right? The electrode is in a specific location in the back. What if you move it a little bit? Then what happens to the preferred stimulus? What happened to the receptive field, right? So if you move the electrode a little bit and the previous cell was responsive to a receptive field here, what do you think happened when I moved the electrode a little bit? What happens to that receptive field? Changes how? It changes very little, meaning that cells that are very close to each other in the brain tend to have receptive field in the same areas of the wall. So, and that's called retinotopy. I say this a lot. We'll talk about that. Another thing you could do, these animals are anesthetized. You could just close one eye and then flash a light and then close the other eye and flash the light and then see what happens. You see that, well, in the surface of the cortex, the inputs are coming from different eyes or next to each other. You could see that when you move your electrodes, the cell becomes selective to different orientations. We'll talk about these columns later on. But the idea is that with neurophysiology, you could actually study these things. You could look at a single cell and what it's doing, especially in early visual areas, because there is a clear connection to the visual world. It gets much harder when you go higher up. Higher up. So again, So a lot of terminology I'm using is with the idea that there's a cascade of activations and processing happening, right? So you heard when people say low level visual areas or low level perception, high level cognition, right? These are kind of assuming that feed forward hypothesis about the brain, that you have early processing from sensory and stuff, and then through a cascade, they become cognitive. Okay, what are some of the benefits of this approach? Well, the pros are that it enables you to localize a certain selectivity to a specific brain cell or area, because you could really lower that electrode and get activation from a very small patch or like few neurons or a single neuron. So you could really get activation that are really localized, spatially very fine. What are the limitations of an approach like this? It mostly can be used in animals, right? Non-human primates are a common model to study. But there are cases where you could do neurophysiology from humans. What would be a case like that? Or like a dead brain?
Yeah.
Well, let's say if we wanted to study uh, an actual live human.
Lesions. Brain lesions.
Could you do neurophysiology on brain lesions? We're talking about neurophysiology here. No, we're talking about neurophysiology. There are different techniques that you could do on humans. I'm asking, can we put an electrode in a human brain? Yeah, so that's a good example. Yeah. So when you have patients with epilepsy, you have to monitor where the source of epileptic seizure is. So you put electrodes in their brain. They actually go to a hospital, they sit there, there are a bunch of electrodes in their brain, and you want to find out what the source of that epileptic seizure is. So you constantly record them. And when they're sitting there, you can actually collaborate with the researcher. They come in and people are bored sitting in hospital. So they participate in these experiments where you show them images or show them different stuff, and you can actually record neurophysiology from them, right? That's a good example of when you can actually record from humans. More recently, there are brain implant companies like Neuralink and others, where there is, they could implant brain in people who are tetraplegic in order to, I don't know if you've seen those videos of, from Neuralink, for example, in terms of controlling devices. And they're recording from the brain. So that's another case where people would sign up for these experimental technologies, where you could actually plant electrodes in people's brain and record them. So another limitation is that you could only record from a single cell or a small number of cells. A small number of cells because the technology has improved since the 50s. The experiment I'm showing you is from the 50s, basically. But more recently, you could have electrode arrays, like Utah arrays, they call them. They have 64, 128 electrodes implanted. Or there are newer technologies like neuropixels that have one electrode, but they have many tips, and you could record from a couple of hundred units at once. But it's like a snapshot of a very small part of the brain, and in that way, it's limited. But you could get a really nice signal from that part of the brain. So some of you mentioned, for example, EEG. That's another technique where you're actually putting the electrodes not inside the brain, but you could put them on the scalp. And some of you may have participated in something like this, but they have to put gels in the electrodes in order to have better conductivity from the skin. And you're not recording the electrical activity of a single cell or a bunch of cells, but you're recording from the scalp. So it has to be a really coordinated activity of a lot of cells in order for you to give you a signal. It's called event-related potential, but the device itself is called EEG, electroencephalography. So the assumption is that if you have a really large signal, so a really large area is coding for the same thing, then it would do, you could actually record it. So for example, let's say you have a person sitting there, you play a sound, and then you could actually look at their EEG signal and see what happens from the stimulus onset. A lot of time, these experiments have to be repeated because EEG signal is very noisy. And then when you average them out, you could get something that they call event-related potential because it's related to this event. And then you're actually recording from the entire scalp, so you could create a figure like this and be like, oh, look, these areas of the activity in the cortex sits underneath the scalp. So these areas of the cortex are more active than these areas of the cortex. So if you have a visual input, you tend to get a pattern like this, where you have like the back of the brain. So this is the nose. So this is the back of the brain, front of the brain. Back of the brain is more active. But it's kind of you could get a general scalp topography map of activations. But the problem with something like this is that it's so coarse spatially. Like it's showing activation of a large chunk of neurons. So you don't really get that sort of really fine-grained signal that you get from single-cell neurophysiology. As I said, the signal is the electrical activity being recorded from whatever is, making it above this count. And also, this is like really tiny signal, by the way. It's not like a big signal. When you average it out, it's a really like microvolts, like very small signal that you have to detect. What are the good things about this method? Well, it's non-invasive. You could use them in humans. I've seen some people trying to build like brain-computer interfaces with these EEG stuff. They're fairly easy to use. They're not as expensive as other methods. The limitation is mainly that they have really poor spatial resolution. That they're not really giving you a fine-grained response, but also they only can detect the stuff that are beneath the scalp. That they're not really giving you deeper stuff. But they have high temporal resolution. So as you get, the signal is really the signal at that moment. It's not delayed. Okay, that's ERP. Moving on to the next one. 10 minutes. All right. So brain lesions. Historically, brain lesions were the first way to actually understand something about the brain. The major assumption here is what? That if you have a lesion to a certain brain area and you observe a change in cognition or behavior, it must have meant that that area was involved in that cognition or in that behavior. Right? If I see a lesion to a frontal area and the person start making poor decisions, it means that the frontal area was involved in decision. So that's the assumption. There are natural lesions like a stroke or tumors. There are concussions that there are a lot of them in like sports, and it's a big topic. There are intentional lesions when I don't know if gunshot. Anyway, the category. So if there's a surgery or a gunshot wound, and then there are animal studies where you could actually lesion yourself, a brain area permanently, or you could just use the type of solutions where you can inject it in there, and then the lesion goes away after a while. Like it kind of freezes the area, so the area is not doing any, it doesn't have any action potential. So you could actually, let's say, inject it with a lot of inhibitory stuff, so the entire area goes quiet, and that's another way of lesioning. It's reversible, using chemical to temporarily deactivate the brain region. All right, so I said that's the assumption. If I deactivate an area or an area is damaged, then it means that if there's a change in behavior, then that area was important for it. So imagine, again, you got the neuroanatomy crash course. This is a very coarse view of the brain. For now, know that, okay, this is the front of the brain. These are where the eyes would be. This is the back of the brain. You could always tell because the front has this dent like that in it. Frontal lobe, parietal lobe, this is the occipital lobe and temporal lobe. Occipital lobe is where the visual areas are, as we talked about it a little. It's in the back of the brain. We have parietal, temporal, and frontal. For now, just this level of anatomy is enough. What people have done is that they looked at all these lesions, and a lot of them are natural lesions, and they kind of mapped out what their importance is. So if you get a lesion to the back of the head, maybe you lose your vision, you become blind, either locally or globally blind. They mapped out these parietal areas that's being involved in body awareness. Um, the the central sulcus is here connecting the parietal to the frontal areas, and then you have the temporal area here. So you could actually use the lesion studies to localize these perceptions. For example, again, in this view of the brain, the front is here. You could always tell that. So this would be parietal areas. And because the person is facing this way, this is the right parietal area. So right here. So people have seen that when there's a damage to this area, they observe something they call visual neglect. And in a way, it's like people are not aware of that part of the world. So, for example, if you ask a patient to look at this, you flash this thing, they're looking at the center, you flash this thing, and then you ask them to draw it. And then you draw it like this. So if they draw it like this, it means that they're neglecting everything in the left hemisphere, left part of the visual world. It means the damage was to what part? To the right parietal area, and it gives them visual neglect on the left side. And visual neglect is different from blindness in what way? Yes, good point, but yeah, no, your hands are not up. How is it different? If I say someone has neglect, how whether they're blind, what could be the biggest difference? Yeah. So in a way, when you're blind, you know you're blind, meaning that when you can't see part of the world, maybe it's black or they're entirely pitch black, and you know that you can't see it. But when you're neglecting, it means that maybe the physical input is coming to you and you could see it in the photoreceptors, but they're not aware of it. They don't know that they're not seeing part of the world. To them, this is a complete view of the world. That's what that means. So you could see them with other paintings, and they wouldn't know. Like they would ask him, "Is this a complete painting?" And they say, "Yeah, that looks good to me. It's a cross. That's a something. That's a house." So to them, they're not aware that they're neglecting part of the world. Um, so this part of the world, when it's damaged, we call it visual neglect, and it's really a perceptual suppression. And the stimuli, like that, it's linked to attention, and we'll talk about attention later, because they're just really not attending to that part of the world. Like they're not processing that part of the world, and they're not aware that they're not processing that part of the world. Okay. Finish this, and then we'll stop here. So brain lesions, based on the assumption that I made earlier, you could use brain lesions to have to infer a direct connection between a brain area and cognition or perception. The limitation is that they're really hard to interpret. Part of the reason is that brain is really adaptive. So when there's a lesion to a certain area, brain can reconfigure itself, right? You could see people born with half of their brain, like just missing a whole hemisphere, but they have a normal life. And some never find out that they were missing a big chunk of their brain. Because brains are adaptive and they can reorganize itself and they could use what's available. And so it's not like that a certain area, if it's missing, especially in early childhood, or if you're born with it, then a lot of time people can compensate for it. Now, when it's lesion, and a lot of these lesions, you just come walk into the lab with that lesion, so you don't really have control or how big the lesion is. We talk about a famous example, which is related to memory later on. But you don't know, you can't really control which part is lesion. You don't know how long it's been. You don't know how much their brain has changed as a result of the lesion. So for all those reasons, although it's been a really good method to get people started to thinking about brain specializations and modularity, it's really hard to interpret because you don't know, but for all the reasons that I said. Okay, yeah, so we'll stop here. We will have to have the next class over Zoom because I'm out of town. You could come here. We're going to set it up here so you could come to the class and watch it on the big screen, or you could just join the Zoom link. I'll send out the Zoom link for the class. But know that it's going to be on Zoom. So if you're not on campus, you don't have to come for it. If you're on campus, you could just come watch it on the big screen in the class. Yes,
When you say positive or negative impact on the cell, do you mean excitatory or inhibitory?
yes.
Okay, great, thank you.
Yeah.